This is Debasish Sinha, Jennifer Salvetti Davis Chair in Ophthalmology, Professor of Ophthalmology, Cell Biology, and Developmental Biology at the University of Pittsburgh School of Medicine, Pittsburgh, Pennsylvania, with an adjunct appointment at the Wilmer Eye Institute, the Johns Hopkins University School of Medicine. In this section, I'm going to talk about abnormal lysosomal function in RPE as it relates to age-related macular degeneration, AMD. So in slide 1, we're showing the patient's view of AMD. AMD, as you all know, is the leading cause of irreversible visual impairment in the elderly. The symptoms vary, but mostly it's blurry spot in the center, distorted images, gray or blank spots. So age-related macular degeneration actually has two different forms, the dry or early AMD or the late wet AMD. The atrophic dry AMD is the most prevalent form, 90% of all AMD cases, for which no effective treatment is available at the present time. So, with the human lifespan increasing, the prevalence of aging-related diseases, including AMD, is also increasing. And despite this growing population of afflicted individuals, no definitive treatment other than the AREDS formulation for intermediate AMD or prevention for dry AMD is currently available. RPE dysfunction has been shown to cause AMD. Recently, Jiang Quan's group from Johns Hopkins University School of Medicine has shown that globally profiled chromatin accessibility in the retina and RPE from AMD and control patients using transposes accessible chromatin using sequencing, or also known as the ATAC sequencing. The chromatin accessibility occur in the RPE with early dry AMD and the retina of advanced disease, suggesting that dysfunction in the RPE drives disease onset. These observations fits with the widely accepted theory that changes in RPE function trigger AMD and suggests that epigenetic changes in RPE cells might be a critical factor that regulates disease onset. Lysosomes are organelles that regulate several functions performed by the RPE as shown in the cartoon. RPE cells are among the most active phagocytic cells in the body, continuously phagocytosing shed photoreceptor outer segments, and as post-mitotic cells with high metabolic activity, the rate of autophagy is also very high. So the end product of both phagocytosis and autophagy has to fuse with lysosomes for degradations. Therefore, lysosomes in the RPE are essential for maintaining retinal homeostasis. Claire Michel's group first showed that the impaired lysosomal acidification might contribute to AMD onset and progression. This was further confirmed by the Recosi group in Australia, who developed an unique animal model for AMD that showed features associated with geographic atrophy. A transgenic mouse line, MCD, expressing a mutated form of cathepsin D, an enzyme that is active only at acidic pH of the lysosomes, and 95% of photoreceptor outer segments are degraded by cathepsin D also in humans, and in these mice this cathepsin D was enzymatically inactive, which resulted in the impairment of the processing of phagocytosed photoreceptor outer segments in the RPE cells. Lysosomes are no longer regarded as only waste disposal organelles. but the hub for several signaling pathways. One being the mechanistic target of rapamycin, complex 1. So the mTOR pathway regulates cell, organ and body size. It interacts with many proteins to form at least two distinct multi-protein complexes, namely mTORC1 and mTORC2. It is a major regulator of both anabolic and catabolic processes. And in a recent National Institute of Aging study, it has been shown that it prolongs lifespan in model organisms. To just go briefly how the signaling pathway works in the cartoon, the proton pump VATPase that regulates pH in the endolysosomal system also interacts in an amino acid sensitive manner with pentameric regulator, a scaffolding complex that anchors the heterodimeric RAG-GTPase to the lysosomes. This leads to the translocation of the inactive mTORC1 to the lysosomal surface. Once mTORC1 is on the surface of the lysosomes, it is activated by Reb. mTORC1 regulates lysosomal function by directly preventing autophagy and the transcription factor TFAB activation. mTORC1 pathway also integrates inputs from major intracellular and extracellular cues to control major cellular events and is a negative regulator of autophagy. Our laboratory for the first time showed that beta A31 crystalline, a member of the beta gamma crystalline family, which is encoded by the gene Cryba1, is a binding partner of VATPase and as such regulates assembly and disassembly of VATPase and amino acids in the lysosomes of RP cells. Thus, it is important upstream regulator of the mTORC1 pathway. As shown in the cartoon, loss of this protein in RP cells activates mTORC1 even during starvation and inhibits autophagy. Next, to provide evidence that loss of beta A31 crystalline affects the autophagy process, we did live cell imaging of RP cells that were isolated from either wild type or mice where we had conditionally deleted beta A3 crystalline from RP cells. These cells were then transfected with pH sensitive reporter mCherry GFP LC3. The idea being in the acidic pH of lysosomes, GFP will be quenched. So these are single frame images in the left side of the slide from live cell imaging to visualize autophagosomes as yellow puncta, since both fluorophores fluoresce at cytoplasmic pH, and autolysosomes as red puncta because GFP is quenched by the low pH of lysosomes using the pH sensitive reporter mCherry GFP-LC3 as I have mentioned. So in the first panel are wild type cells. Since the wild type cells lysosomes are acidic, the GFP is quenched and you can see mostly they are autolysosomes as shown in the graph. Next, in the conditional knockout mice RP lysosomes, we have shown that the pH is no more acidic, so GFP cannot be quenched, and so you can see yellow, more of yellow dots that indicating that there are more of autophagosomes. If this is true, to confirm, we took again wild type cells and treated them with baflomycin A1, a known inhibitor of VATPase and lysosomal pH. So again, GFP is not quenched and you can see that there are more autophagosomes than autolysosomes. To prove that this is happening because of the loss of the protein beta A31 crystalline, we took the conditional knockout RP cells and transfected them at overexpressed beta A31 crystalline, and what we find that the pH is retained and then the GFP is quenched and you can see that there is more autolysosomes than autophagosomes. The autophagy defect in Cryba1-RPE cells does lead to age-related macular degeneration-like phenotype. As shown in the left-hand side of this slide, RPE flat mounts from both wild-type and knockout mice show that with increasing age, RPE cells in Cryba1 knockout mice gradually lose their regular hexagonal shape shown by arrowheads and have less RPE65 shown by arrows, a marker for RPE cells. Also, abnormalities in the cellular architecture of Cryba1 knockout RPE is being shown by the transmission electron microscopy. In knockout mice, large basal laminar deposits in Bruch's membrane has been shown in panel F in asterisk. A hallmark of development of AMD could be seen. Defects in lysosomal-mediated clearance also affects retinal function. As shown here, ERGs revealed impaired retinal function that increased over time in conditional knockout mice. At 4 months, conditional knockout mice and the flux control mice showed no significant difference in scotopic A wave amplitudes at any stimulus intensity, suggesting that rod photoreceptor function was still intact, as shown in panel A. But CKO mice showed a significant reduction in photopic B wave amplitudes at the 2 highest stimulus intensities, indicating cone photoreceptor dysfunction, as shown in panel B. At 7 months of age, CKO mice showed significant reductions in both scotopic A wave and photopic B wave amplitudes, indicating dysfunction in both rod and cone photoreceptors C and D. Since cones constitute only 2% of the photoreceptor population in mice, gradual loss of both rods and cones would result in a greater percentage loss of cones than rods at early time points, and hence loss of cone function would be identifiable prior to rod dysfunction, as was reported. Interestingly, other mouse models of AMD, such as Sod2 knockdown and ApoE mice fed with high-fat diet, demonstrated an age-related increase in autophagosome numbers and expression of autophagy proteins. However, Michael Boulton's group also found that autophagy proteins, autophagosomes, and autophagy flux were significantly reduced in these 2 animal models of AMD. They concluded that autophagy plays an important role in protection of the RPE against oxidative stress and lipofuscin accumulation, and that impairment of autophagy is likely to aggravate oxidative stress and contribute to the pathogenesis of AMD. Their study also showed autophagy is dysregulated in the RPE and neural retina of human AMD specimens. Moreover, David Sachs' group elegantly showed that deleting selectively RB1-CC1, a protein essential for induction of autophagy in RPE, resulted in multiple autophagy defects within the RPE with decreased levels of lysosome markers as shown here and defective processing of intracellular debris. An elegant study from the laboratory of Nady Goldsteiner showed dysfunctional autophagy as an underlying mechanism contributing to the pathophysiology of the disease using cultured human RPE from AMD donor eyes. Their study showed that the ratio of LC32 to LC31 revealed impaired autophagy in AMD RPE as compared with normal RPE. Autophagic flux was also reduced in AMD RPE as compared with normal RPE, as shown by inability of AMD RPE to downregulate P62 levels during starvation. Another study done by the Health ABC group, led by Alessandro Iannacone, showed a role in autophagy activation by all 5 autoantigens as shown in the graph, raising the possibility that the detected autoantibodies by ELISA may play a role in AMD via autophagy compromise. Based on these animal models of AMD and from human AMD patients, we can conclude that aberrant lysosomal clearance function of RPE can lead to dry AMD. Rejuvenating lysosomal function in the RPE can prevent or delay the progression of AMD at the early stages of the disease, and inhibition of the mTORC1 pathway can rescue the abnormal lysosomal-mediated clearance in RPE and thereby have a preventive and therapeutic effect in dry AMD.